Good morning, my dear students. I am Dr. Jitendra from Department of Biotechnology and Sras Mahila Mahavidyalaya. So the topic for today's lecture is we will study the production of material production of microbiological production of cheese, which is a very important fermented product and part and parcel of our daily life. तो आज हम इसका सारे इसकी माइक्रोबायोलॉजी और starting from the raw material, what are the various steps involved and how it is industrially produced. So we will study today about this very important topic that is the of industrial microbiology, that is the production of cheese. So cheese is an integral component of our life. We are consuming in our daily life also. So my dear students, you are also very well versed with the different types of the cheese. Today we will discuss how exactly these different types of the cheese they are produced and what are the different processing techniques which are involved in order to produce one and other kinds of the cheese. So I am just starting with the topic, that is how this cheese can be produced at industry level. So all of you know the production of cheese involves the raw material which is used for the production of cheese is the milk. All of you know very well first. So milk से हम जो है इसको बनाते हैं, milk is the raw material. So there are basically 4 steps which are included in this whole process, which we will discuss term by term. So I am just drawing first of all the kind of the process which is used in the manufacturing of this cheese. So this is our layout for industrial production of the cheese, and I have written some of the important intermediates which we are getting in this process, and I will explain one by one all these steps. So my dear students, this is the layout for the production of cheese, which is a fermented product and part and parcel of daily life. And first I will explain this very first step, that is how this milk is changes into coagulum. So first we have the bacterial culture. So this bacterial culture, we have inoculated the milk with the bacterial culture. And first I will give you the composition of the milk. So the composition of milk is concerned, it consists of the sugar lactose. Beside lactose, it contains fats, lipids, and it contains the unique protein which is called casein, very important protein, and then we will have beta carotene. So these are the important components of the milk which we are taking into consideration the formation of cheese. So this beta carotene is a very important pigment, and this is present in the milk, and lactose is the sugar, fats and casein, they are the components. So first step is the bacterial culture. So what it will do, it will change the lactose into lactic acid. It will change the lactose present in the milk into lactic acid, and due to the presence of this lactic acid, the pH of the milk goes down. That is, the pH of milk, it will be now about 4.6 around. It becomes this light acidic. Otherwise milk is having pH around 7. But due to this bacterial culture and conversion of this sugar lactose into lactic acid, pH of the milk slightly comes down and it becomes the acidic. Then second very important is the addition of rennet. So in this step also, so rennet is added. Rennet is an enzyme which is a very important enzyme, and this rennet will do the hydrolysis of casein. Now the casein, I would like to discuss something about the casein first, then you will understand the meaning of hydrolysis of casein. Basically milk is a colloidal solution, and this colloidal nature of this milk, it is due to the specific kind of the protein which is called called casein. And this casein protein, if we analyze this casein protein, then we have seen that it has two regions, C-terminal region because it is made up of amino acid, and N-terminal region. So C-terminal region and N-terminal region. So these are C-terminal region of this casein proteins, which is hydrophobic in nature, and which is a lipid loving basically, and this N-terminal region is hydrophilic in nature, that is water loving. So because of this presence of this hydrophobic and hydrophilic moieties in the protein of this casein, so this casein protein is not soluble, so it remains in the suspended form in the milk. So this is basically the how the milk is colloidal solution in nature, and it is due to the presence of this unique protein which is called casein. So casein is the protein which, and this rennet will cause the hydrolysis of the casein. So इस hydrolysis का क्या meaning है? Hydrolysis means that this rennet will act on this casein and separate both hydrophilic and hydrophobic regions, and that is why this casein now will be precipitated and it will settle in the bottom, right? So that is what you mean by coagulation of milk, and coagulum formation takes place. But इसमें सबसे important जो point है वो ये है कि hydrolysis of casein it is only taking place in the presence of calcium. So now question comes, from where this calcium will come? So calcium is already present in the milk, there is no problem, no addition of calcium will be there, and this rennet will automatically changes or hydrolyzes this casein into hydrophilic and hydrophobic region, and coagulation of this casein will take place, and the resultant product form is the coagulum. So this is very important step. So how exactly this? And third very important step in the formation of cheese is the addition of lipase. So lipase will bring about the digestion or hydrolysis of the lipids. So again, these are but lipase will be added. The important thing which needs to be taken care of is lipase must be added before the rennet is added. And एक और इसमें बहुत important step है कि this is very much temperature dependent process. rennet जो है rennet casein के ऊपर जो act करेगा, it is very much temperature dependent process, and it takes place at around 32 degrees centigrade. This is very important point. And second very important point is the presence of calcium. So in that case, when these three things will act on the milk, which is the raw material in the production of cheese, then the resultant component is the coagulum, right? So this coagulum, after the coagulation formation, then it is slightly, then the process is called cooking. Then the process is called cooking. Cooking, as its name suggests, ये नाम ही बता रही है कि इसके अंदर we have to heat this coagulum at a temperature 37 degree temperature for a certain period of time. And when we heat this coagulum at this temperature, then this coagulum will result in the production of two very important components which can be separated automatically from each other. And what are these products? So the solid product which is produced at the below it is called the curd, and the watery liquid which comes on this curd, that is called the whey, and it is separated. And this whey, since it is rich in the protein component, so it is again used for some other purposes, and this curd or this component is further used for the production of cheese by taking into consideration the different processing steps. And with the help of this processing step, we are results in the formation of the cheese. So अभी आपको कर्ड का पता लगा है कि कर्ड और whey milk से कैसे convert हुए. These are the steps which I have explained. Now I am coming to the processing, that is the conversion of curd into cheese. कर्ड into cheese बनाने के लिए हमारे को जो सबसे important step है, first step is the salting. What we are doing at industrial level is the salting. तो salt इसके अंदर हमें ऐड करना पड़ेगा, and salting all of you know it plays a very important role in terms of antibiotic microbe in nature, and it is acting as a preservative. So curd, first it has to be done salting, and then cheese bricks, and this curd is now after inoculated with certain kinds of the specific kinds of the microbial strains, and or sometimes we are also adding enzyme also like lipase, like protease. So these are the enzymes or microbial strains. So these cheese bricks which are formed after the conversion of curd, तो जो इसके अंदर फॉर्म होगी चीज ब्रिक्स, they are specifically inoculated with the specific kind of the microbial strain. So why exactly this thing has been required? Because the thing is, when we are adding the enzyme protease, for example, what the protease will do? Protease will definitely do, it will cause the breakdown of proteins, right? So proteins will be broken down, and smaller peptides they will be formed, right? And lipase it will bring about the hydrolysis or breakdown of lipids, right? And in this case, lipase it results in the production of butyric acid. So butyric acid is a organic compound, so the component which is strongly responsible for flavoring agent. Flavor के लिए butyric acid, which is produced by the action of lipase, it has been used. So it depends upon the kinds of the cheese which we want to produce. So accordingly, we have to inoculate these cheese bricks with the specific type of the microbial strain, or with the specific type of the enzymes. So this is very important. This is very important aspect. So for example, I am giving you one example. If we want to produce the cheddar cheese is produced. So we have to inoculate the cheese bricks with the specific strain of Bacillus amyloliquefaciens. So this Bacillus amyloliquefaciens, this is the very important liquefaciens bacterial strain, and it is produced particularly. For example, I have given you one example. Likewise, in order to produce the different kinds of the cheese, we have to inoculate these cheese bricks with the specific strains. So in that case we are producing. And one more important thing while processing we are doing is, processing में एक और चीज इसमें बहुत important है कि processing, for example, attributes of food is very vast. We are going to produce so many kinds of the cheese which differ in terms of like aroma, texture, flavor, and consistencies, mineral values, and all these things. But one important thing is, if I take into consideration the flavor, for example, then protease. What exactly the role of protease enzyme is very crucial and very important. So how exactly this protease will do? Protease obviously they will brings about the hydrolysis of the protein, and smaller peptides they are produced. So if the N-terminal region, if these peptides they have N and C terminal regions, if the N-terminal regions they are rich in acidic amino acid, from where these N-terminal regions peptide short chain has been created, they are generated from the protease enzyme. So if they are rich in acidic amino acid, then it has been found that they are producing the meaty flavor. मीट जैसी फ्लेवर आएगी उसके अंदर, तो जो आपका चीज होगा उसके अंदर वो फ्लेवर आएगी, और accordingly the objective or which we want to produce, that is there. Similarly, if it consists of another types of C-terminal region if it is having basic amino acids, then obviously we have the different types of the flavor is generated, and bitter flavor is there. So this is one example how exactly we can introduce or we can process the curd into the different types of the cheese. So cheese basically is, all of you know, it's of great variety, it's of great different types. It differs according to so many parameters which I have already discussed you. Just like जैसे मैंने आपको अभी बताया है कि चीज़ में कितने टाइप्स हो सकते हैं चीज़ के. सो चीज़ की कंसिस्टेंसी क्या है, अरोमा क्या है, उसके अकॉर्डिंगली हम इसकी प्रोसेसिंग करेंगे. एंड प्रोसेसिंग इज वेरी मच साइंटिफिकली इट हैज बीन वेरी एस्टेब्लिश्ड स्टेप्स. So this is we are producing it successfully. There is no any kind of issue in this process. This technology is standardized technology. So we are getting this product. And in the end, I would like to add some important thing here is this is a kind of a fermented product. Why we call it as a fermented product? Because fermented products are all those any process in which the biological agents, which may be the microbial cell, which may be the plant cell, which may be the animal cell, they are involved, and in which a product of economical value is obtained. So we call it as fermentation in broader sense. Otherwise, fermentation has different definitions in terms of metabolism, in terms of biochemistry, in terms of industrial microbiology. But this is one definition in terms of industrial microbiology. So industrial microbiology, we are getting these fermented products. So I will call it as a fermented product because it is, all of you know, this product has been exclusively produced as a result of so many microbial activity. Whatever I am considering lipase, protease, bacterial culture, red net, everything is there. So I can say that this is a fermented product, and fermented product is having a large number of advantages over the non-fermented products, which I will be discussing in some other lecture. And one more important thing I would like to discuss over here is the the nutritive value of the milk and nutritive value of the cheese is quite very different, because ultimately it is produced since the activity of microbial activity. So this is having more nutritive value as compared to the milk, I can say. Otherwise, both have its own importance, both have different kinds of the composition in terms of the chemistry. So इस टाइप से हम different products जो है industrial microbiology के अंदर produce कर रहे हैं, and successfully we are exploiting these microorganisms for a number of the products nowadays, and technology has been standardized and we are using in our daily life also. They have a great impact on our health as far as well as the economy is concerned, as well as the other sectors are concerned. So this is what about the production of cheese. So in the next lecture I will discuss about the production or the making of some another fermented product, and I think hope you will get the basic idea about this whole process and about the product which we have produced. So thank you for patience listening, and we will shortly meet in the next lecture for another fermented products. Thank you very much.